I love this conference, and I'm really thrilled to have been invited to share with you some tools we're developing that I hope they're about to go out into the wild so that we can do human immunology instead of all the mouse immunology we do to try to cure endometriosis. And so I'm going to focus on one application, but the tools I'll talk about are very general. And to anchor this, I'm just one historical slide that connects me very firmly from my Cambridge to this Cambridge, is that early in my career I worked in regenerative medicine, building scaffolds for tissue engineering. I translated a lot of things into the clinic, co-inventor on the original 3D printing and so on. And I had an epiphany though in working with a liver transplant surgeon that wouldn't it be better to cure liver disease instead of doing liver transplants? So we've embarked on an effort to create living human models of liver that could be used in preclinical drug development, particularly in toxicology and so-called ADME, drug metabolism. And I've worked with pharma companies, about a dozen of them over the past 30 years. Every single problem they ever brought to me to use our little 3D liver bioreactor involved immunology, things like idiosyncratic toxicity, and so on. So many people liked our original liver chip, which was literally a silicon chip that we could grow 3D liver in with pumping. Pumping is very, very important in these systems. We translated it to a multi-well plate format using some very clever pumps that involve air and vacuum to drive pneumatically an onboard pump with little membranes and fluidic channels. And you'll note, these are not the cute little chips that you often see, the little PDMS things. They're made out of a hard plastic because in drug development, most drugs are highly lipophilic and they will absorb into silicon rubber. So you have to have something that allows you to do PK. So this, around 2009, finished, and then we translated this to a company in Cambridge, CM Bioinnovations. They made a very nice commercial version that we use, and many drug companies all over the world use this, and the FDA has used it and validated it and so on. And we have many collaborations with pharma continuing to use this version of the liver chip, which is very simple, but it keeps liver function for a very long time in culture. So you can do many things, and you can include immune cells and so on. At the end of the talk, I'm going to come back to our new technologies, because the focus of today's talk is going to be on developing new tools that will become accessible to you. Some of them are going to become accessible this summer. And so my role at this system is to help us all do better studies of women's health. So around the time we were getting the liver chip into the multi-well plate format, I had a bomb drop in my life. And that was my beloved niece who grew up in Atlanta, started her period, and she had terrible problems, terrible. Did not respond to birth control pills. She had tons of GI problems. Tests showed she had no cancer, this and that. So the doctor told my sister, your daughter is making everything up to get out of going to school. Okay? I got her referred to a surgeon in Atlanta. She had stage 3 endometriosis when she was 16. Okay? So my head exploded. She was an amazing student. She won all the awards, got multiple full scholarships. She loved school. Why, in the 21st century, is a female doctor telling my niece that she's just being hysterical? This was crazy. So what happened then is I got an unexpected fellowship that let me have some mad money and also a foundation, and I got pulled into doing research on endometriosis, founding a center at MIT, which is mainly an engineering technology school, with a fabulous surgeon, Keith Isaacson, who's very involved in running the big professional societies and so on. He says, he said, you know, I operated on a 23-year-old girl. It was her 10th surgery for endometriosis. We need engineers in this field just to bring new perspectives. Can't you get all these smart people at MIT to work on endometriosis? Now this is very scary because you know what the funding levels are. I was in a field very well funded, so to jump in something I didn't know was a bit scary. And so I brought a fabulous MIT collaborator, Doug Laufenberger, some systems immunologists may know him, into this, and we decided to really take a deep dive into endometriosis. So we've had a little bit of intro to this. It's a very common chronic systemic inflammatory disease, and it's treated right now with estrogen suppression therapy. So you can take birth control pills, which have side effects. You can most common second line is menopause-causing drugs, and patients who fail those will have surgery sometimes, 5, 10 times and still not get relief. And this is an operating room where I collect samples myself. I go there sometimes to observe surgeries to understand the dimensions of the disease. So I wouldn't be talking to you if there were great drugs or great ways to develop drugs. Pharma has avoided this, and my job is to make all of you have better tools so that pharma can have lower barriers to going even into this space at all. Now, it's a really interesting disease in that the lesions form, they're bits of the endometrial tissue, or like tissue, growing in the abdominal cavity in these lesions that, of course, attract a lot of immune cells. The theories of why it formed are highly debated. Is it developmental? Is it reflux menstrual tissue? Coffee breaks, bar? I will fill you in on all of that. But a point is, you can find lesions everywhere, but they have an unusual tropism to very stiff tissues. So you find them invading the diaphragm and getting to the lungs, invading the bowel wall. And you can find them also in the actual myometrium, and that's a disease called adenomyosis, the sibling, if not the twin, of endometriosis. And look at this beautiful polarized epithelium sitting in muscle. Carcinoma doesn't even do that. It's crazy, isn't it? And you can see, and this is just a slide one of the postdocs in the lab did once upon a time, it's got immune cells in that epithelial layer. Now, of course, given that the incidence of adenomyosis is probably comparable to endometriosis with similar symptoms, you'd think there'd be a lot of intensity of research on this. But if we use PubMed as an integrator for research all over the world, we can see for adenomyosis, compare it to a disease with comparable morbidity, and there's only about 5% of the research intensity. This is crazy. It's also an opportunity. All you young people, I'm here today to say, go for it. And I will help you develop the tools. Okay, now, a good bad thing happened right after we started our center. I got diagnosed with triple-negative breast cancer, and it was really interesting because I found a tumor myself in Singapore on a Friday, and on Monday I had, you know, the biopsy and blah, 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 and on Wednesday I knew I was triple-negative. So there's 3 canonical markers, ER, PR, and HER2, and that was related to the mechanism of the disease, the prognosis, and the therapy. And so here's what's going to happen. The oncologist did not say, Linda, your tumor's 3 centimeters. Here's what we're going to do. No. It's gated by, well, you're going to have supersized chemo because there's no targeted therapy. But for endometriosis, the disease is classified by lesion burden. Okay, but 10% of women have this. Of course, there are likely molecular subsets. So when we came into this, it was the aha moment. How do we find those molecular subsets? So it's been a long journey, and this is really setting the stage for the tool development, because I do want to tell you something that we found instead of just stuff that we did that you're going to use to find stuff. So the first thing we did in endometriosis is just get our own sense of the disease by studying the peritoneal fluid. Now you can study the eutopic endometrium, because it's deranged. You can study the peritoneal fluid where there's known to be a lot of derangement of immunology, and you can study lesions. And we've done all of this, and I want to just set the stage again by showing one of the first studies we did, and this was in collaboration with Doug Laufenberger. We took peritoneal fluid, and this is back in 2010, and did profiling of 50 cytokines with Luminex in about 100 women, about 20 controls, and the rest patients. And then we asked, you did a multivariate analysis of this to ask, are there cytokines that go up and down together, suggesting a network? There were in about 1/3 of the patients with all stages of the disease. And when we deconvoluted the immune network and did some bioinformatics to look at transcriptional control of cytokine production, it led to a certain type of macrophage in these patients and Jun kinase controlling the inflammation. So this was very exciting, okay, because we now found something other than an estrogen suppression target, potentially Jun kinase, controlling some of the features of this. This was the first paper describing a molecular classification of endometriosis. It's not a perfect one, but it got the field thinking that there should be molecular classification. And keep in mind, there are not somatic mutations that are known to be drivers. There's a lot, I can talk about somatic mutations with you at the break if you want, but at this time, looking at the protein networks and network states were the way forward. We did a companion study on invasion, and this is a patient of Maurizio Brau, a fabulous surgeon in Sao Paulo, with whom we collaborated, and this patient had 2 huge lesions in her bowel. She couldn't poop. So she had 10 centimeters of her bowel excised. In this study, which Doug Laufenberger led, and we invented a new way to study protease networks, some tools for that I won't talk about, we also identified Jun kinase as a major driver of phenotype. So this was really exciting. Maybe we will have Jun kinase as a driver. So we published these, and then we discovered that a local drug company, Merck Serono, had been trying to develop Jun kinase inhibitors for endometriosis, and in fact had cured multiple patient populations as noted here. Now, they licensed the drug, because big companies don't like to do endometriosis trials, to a small company, and the clinical trial was not successful. Some of it had to do probably with trial design, and some of it may have to do with the nature of the particular molecule they used. But nevertheless, the Steve Palmer, who drove this at Merck, I ran into him at the American Society for Reproductive Medicine in 2014, and he was explaining to me what he thought needed to be done to get Jun kinase inhibitors into the clinic, because he really, really believed in it. And he's a very quirky and very dedicated person. So there was about a foot of rain that weekend in Honolulu because there was a hurricane. So we spent a lot of time talking about the future. And what he told me, he says, what we need is we need human efficacy models. We need a little lesion, like what the surgeon would leave behind, with blood vessels that can attract the immune cells. That's what we need to test our drugs on. So that's what he tells me. I'm like, okay, let's see what I can do. So Steve went to Baylor to develop new Jun kinase inhibitors. That's how much he believed in them. He went to Baylor. He says, I'll develop new molecules if you get the preclinical models. Okay, so what did we do? Did I X out some very important? Yeah, okay, so we started to study the immune cell. We need to build these. So if we want to build the lesions, we need at least the epithelia and the stroma. And so you can collect these from biopsies from patients, and we do this all the time. We have very, very dedicated clinical collaborators. I can tell you the MIG surgeons love having scientists come to their meetings, and if you just go to their meetings, they will happily help you collect samples. So we started by building a tissue bank. We wanted to build it, how are we going to do this? So we needed a lot of tools, and we needed the primary cells because the cell lines don't really recapitulate. So as you probably, most of you in this room know, progesterone, when you give it to cells in culture or in the body, when you have a surge of progesterone at ovulation, you induce the decidualization response, huge changes in gene expression. In culture, it's easy to track it with a surrogate marker, production of prolactin in stromal cells. Okay, so when the stromal cells become epithelial-like, they make basement membrane, gap junctions, and so on, very characteristic changes. But a lot of gynecological diseases have a phenotype called progesterone resistance, where they lose their sensitivity to progesterone, and it's often the stromal epithelial signaling that drives the phenotype in the endometrium and all the changes, so you really need the stromal cells to respond. Okay, so that's an important phenotype. Now, if you take samples from a lot of patients, these are just about a dozen patients in our tissue bank, and you measure prolactin as a surrogate, you can see the ones in blue are our control patients. There's a lot of variation in the response, including some of our endometriosis and controls look sort of similar. And the controls in our study may have fibroids because we're not asking women to come in and paying them to do a biopsy. So we're learning a lot about the spectrum of this. But certainly these two patients have a very strong progesterone resistant phenotype, and their clinical phenotype is consistent with having lots of inflammation that would drive this. And so I'm not going to have a chance today in this talk to talk about all the things we've learned. I am going to focus more on tools that you might want to use, but these are the kinds of studies or patient samples that we have. Now, when we started this work, after Steve dropped that bomb on me, we only had cell lines, or you could do experiments at one sitting with epithelial cells, because there were no ways to grow the epithelial cells to make a tissue bank. When Hans Clevers showed that you could grow mucosal epithelia in Matrigel with a cocktail of growth factors, this, of course, revolutionized all the studies of mucosal barriers, because now you could take a single biopsy and expand it. So soon that work was expanded to or applied to endometrial organoids, and these are the two seminal papers describing somewhat comparable ways to do that, and variations of these are what we use and what everyone pretty much in the field uses, although to make organoid cultures and then freeze them down so you have tons of samples from an individual patient. Now, One of the problems with the organoid cultures is they are grown in Matrigel, which has an enormous number of problems with respect to doing biology. Number one, it comes from a tumor, and there's all kind of stuff in it that interrupts signaling. You have to have a TGF-beta inhibitor. You can't do co-cultures very well because the cultures just break down, the stromal cells overrun, and on and on and on, lot variability. So the first thing that we had to do, I come from a biomaterials background, so we spent some time developing a completely synthetic replacement for Matrigel to culture organoids, okay? And it's a very simple conceptually, but devil's in the details. So we have a synthetic polymer made of polyethylene glycol. It has some peptides on the ends of this polymer that interact with cells, with integrins and other things. It's cross-linked with a peptide that can be cleaved by MMPs. So cells can remodel this. You mix these constituents together and have a little shift in pH, and you do the cross-linking and get a beautiful transparent gel, as you see here. And cells are perfectly fine with this. They survive. It's a very gentle process. Okay, and again, the devil's in the details. It took us about 10 years to develop this. There's an enormous amount of engineering that went into this, and it will become commercially available later this year. The company is Biotechne will have it. It's in the process of beta testing, and some people in this room have used it, and we do want to share it, but it's not like sending a plasmid. It's actually a lot of work for us to send this out, so that's why we're happy Biotechne is going to let all of us order it out of a catalog. To go a little more into the details, not hugely into the details, but there's some features that are useful. When you put the cells in the gel, they're naked, okay? They don't have any adhesion molecules. They need to have adhesion signaling. So the first step in this is to put peptides in that will bind to critical integrin ligands that the cells have. Okay, so that tells them, okay, you're gonna be okay. Here's a little blanket for you. And then once they're there, they start to make their own matrix. And an epithelial cell will make basement membrane. So laminin, heparin sulfate proteoglycan, and collagen 4. The stromal cells, they'll make a fibronectin, transitional matrix, and so on. And immune cells, we put immune cells, and you'll see, and so on in here, and we publish on all of this, so I won't go into all of it. But what we did is we very carefully tailored it for those two stages of biology. Get the cells to be happy, and let them go off onto their path where they can remodel their environment and make a tissue-like structure. So that's the simple thing, and there's a lot of little rules that you have to pay attention to. Now, these are just some gratuitous images of organoids growing on the left in Matrigel and on the right in the synthetic gel, and a little cheat. These are actually gut organoids because in the US you can just open a chute in your ceiling and money falls in your office to work on gut, but that doesn't work for endometrium. There's no endometrium chute. So we got a lot of money to develop this for the gut and then recycled it to do endometrial organoids, of course. And so this is from a paper published a couple of years ago from a fabulous postdoc, Juan Neco, who's now at Tufts, where we can encapsulate the cells, stromal and epithelial, together. And this is just showing some morphologies of the cells going through a simulated menstrual cycle with different hormones. And we use mifepristone to inhibit the progesterone to simulate menses, just make sure we get everything out. But you can see that there's a very nice prolactin response, and then that goes away when you take away progesterone. And so there's a lot more details in the paper, but here you can see they're beautifully polarized. The actin is apical, and the red are stromal cells, and they like to migrate and hang out with the epithelia. Now, we can start to model facets of endometriosis with this by putting in an inflammation cue, something like IL-1 beta. Now, IL-1 is a double-edged sword because it's essential for certain endometrial functions like implantation, but too much is involved in endometriosis. And you can see right here the huge morphological changes with inflammation, the flattening of the epithelia, the excitation of the stroma, and we lose that responsiveness to progesterone. So this is the control, and this is the inflamed, and they lose responsiveness to progesterone as you would have in a patient with progesterone resistance. And this inflammation was already a known cue to induce this phenotype in vitro, but now we have crosstalk. And the beautiful thing about this for biology, and we have a paper with Hillary Critchley about ready to submit that I don't have time to talk about, looking at the crosstalk in samples treated with ulipristal acetate, where there's some concerns that drug's not approved in the US because of changes in morphology. And the synthetic matrix much more faithfully captures the changes that she sees in patients she has measured changes in than Matrigel co-cultures do. And it's really, really striking, and this is probably because there's no interfering signals between the stroma and epithelia in the synthetic matrix, so we don't have any proteins in there that would signal other than the integrin ligands. So one nice thing about a synthetic matrix that you can control the properties is we think about multiple cues influencing endometrial function and all of tissue function, not just the biochemical cues that would arise from which integrin ligand do you have, or did you have progesterone or not, or IL-1 beta or not. There's also a lot of biophysical features of the microenvironment that may dictate response. And in the case of endometriosis, the figures I showed you before, you see lesions in very stiff microenvironments. And there's been tremendous interest in the biomaterials and cancer community, and does stiffness dictate function, stiffness of the matrix. And I want to be very careful because you can often not deconvolute stiffness from other biochemical effects, but it is one of the parameters that can control this. Now, this is a lesion, adenomyosis lesion. Keith Isaacson, our collaborator, is really, really into adenomyosis, thankfully. So you can see the lesion on the left, and then you see a light sheet image of that lesion showing the epithelia invading into the muscle. So it's kind of like fracturing the tissue almost, and that's a fluid-filled lumen there. So we can debate at lunch. I'd love to talk about why they can do this. It's maybe hydrostatic pressure because of fluid transport imbalance, et cetera. But what we can do with the synthetic matrix is we control the cross-linking and biophysical properties of this matrix. So if we grow the cells, and these are co-cultures on the left, are cells in a very soft matrix like the endometrium. On the right are cells in a stiff matrix like that of the myometrium. And you can see they take on this invasive phenotype. I'm not going to show you the movies, but they also make inflammatory cytokines at a much higher degree, pro-inflammatory cytokines. So these are things that we're continuing and have a lot more data on that I won't present today, because I do want to share tools that we may be able to get out into the wild that you could use for your own studies. Okay, another phenomenon that we observed, and it's a little hard to see because it's light here, is that when the organoids would get close to the surface of a synthetic gel, they would actually emerge out and form a monolayer. So you can barely see here, there's some organoids, but there's a green haze, and that's because they grew along the surface of the gel. So now you have something, you have something like, so schematically here, if we look at two different focal planes, on the surface there's a monolayer, and interior, showing lamin and basement membrane, you have a gland-like structure. So we're capitalizing on this, and we have an NIH grant with Rosera Ventatormo, who will be speaking this afternoon, to study this fascinating phenomenon and benchmark it against her beautiful in vivo molecular and cellular studies across the cycle, and we're focusing on the late proliferative to early mid-luteal phase in that work. But I want to come back to endometriosis and finish there, because a signature thing about all these lesions is they're extremely highly vascularized. And our colleague Roger Cam at MIT over decades had invented a way to take a microfluidic device that has a fluid channel on either side of a central compartment that has little posts that allow you to inject a fluid into the central compartment that can form a gel. So think fibrinogen and thrombin, and you put those in, they're held in by surface tension, they form a gel, then you can come back and fill with fluid. So if you include vascular cells in that fibrin thrombin, you'll form a little tissue compartment that over 4 days turns into perfusible microvasculature. And you can see here, this is driven by gravity flow, immune cells flowing through those vessels. And this is a commercial chip, and it's made from a hard plastic, so you can put steroids in it. Okay, so we actually use this for a lot of our work. So we got a joint student with support from NIH, and Ellen adapted this approach using a PDMS chip as a prototype here, where you can put a little hole in the vascular bed as it forms, and then, because this is fibrin and thrombin, fibrin bed, and you can come back and put a proto-lesion and watch blood vessels grow into it. And what we have data on now, and she's finishing her thesis, are beautiful differences between progesterone-resistant and not progesterone-resistant tissues with respect to some of the angiogenesis markers. But we really, if we're going to translate this out into the wild, especially if we're going to work with pharma on drug development, we can't really use fibrin. We really need to have everything in the synthetic hydrogel. So a fantastic postdoc in the lab, Lauren Pruitt, together with Priyatanu Roy, developed a way to make the vessels through in a synthetic gel. And this is the first time there's ever been perfusible vessels in a synthetic gel. So on the right, and it may be a little too bright to see very well, we inject the proto-lesions together with the vascular cells, and these are uterine vessel cells in this image, and over 6 days the vessels form, the lesions grow, and they become vascularized. And so now we're developing a lot of imaging to look at the metabolism and structure in this using label-free imaging. I won't show today, but happy to show slides otherwise. I'm going to finish with a couple of challenges we're working on. Back when we started with liver, a huge problem was controlling cortisol concentrations, because most liver culture would use like 10 micromolar cortisol, when free cortisol in your plasma is like 50 nanomolar. So we really got into the fact that all your steroid hormones are mostly bound to plasma proteins. CBG binds cortisol and progesterone, and SHBG, estradiol, and testosterone, and you really need these in culture if you're going to control steroid hormone levels. You want to have, like I was very struck by the PCOS, you have kind of constant and then normal is like this. We'd love to create our microreactors to do that kind of control. It's a very tedious process. We just got a few $1 million to do this. It's really complicated to do it, and we're working with the group in Edinburgh who are really experts in this. So that's a tool that'll be about a year down the line at most, because you can't get these recombinantly. You have to make them yourself or get a company to make them. So that's one of the things we're working on, not just cortisol, but obviously all the steroid hormones. And I'll come full circle. So Steve Palmer got some very exciting new molecules. He licensed them to Cellmatics, a startup company out of New York. And we now are working with him to test the new Jun kinase inhibitors in our model. And Jun kinase inhibitors have pleiotropic effects because Jun kinase is in a lot of cells. And so it's very important to look in the context of cell-cell crosstalk dynamically to do this. This is the team. On the left are the Cellmatics folks, and on the right is our lab. And of course, Doug Laufenberger is a complete collaborator in all of this. And I'll end by saying that at MIT, we are committed to building a living patient avatar core facility. We've moved far from the liver chip. We have a whole new platform for these little tiny chips, and I have a little example I meant to hold up, but I left it in my purse. But we've got the lesions, we're doing pain neurons, we've got a vascularized liver model, because if you're going to go in the clinic with any drug, it's got to be tested in a female liver. And Bayer's really great potential new endometriosis drug failed 2 years ago in phase 2 clinical trials because of liver toxicity, after I told their scientists that they should test it in a female liver, and they didn't listen to me. They're men. And then we have a very fascinating story with Mickey Tall about Lyme disease, chronic Lyme disease, which is far more prevalent in women. And she's got a paper that she's just submitting showing that prior history of infection with Lyme potentiates you for certain gynecological diseases. So we're building models of this in the lab, and it's totally wild. And then we have a gut device. So I'll end there and say huge thanks to the big team of people, especially to Doug Laufenberger with his first student who worked on endometriosis and the cake the lab made her that's like a uterus. Dave Trumper, my hardware collaborator, fabulous clinical collaborators, and lots of folks. And the new collaboration with Rosa Ventatormo is really getting me to understand genetics. Thank you.
Thank you so much, Linda. That's great. I think we've got time for one very quick question, if we're very quick. Yeah, back here.
Yay. Great talk. I was wondering about this invasive phenotype in the stiff matrices. Is there any bimodal response? So how stiff is stiff, and is there differences to what happens in between? And I think your matrix is biodegradable, right?
Yes. Okay.
Do you
so
control
the question
for
is
attachment
about the
size?
stiffness. Is there a dose response essentially of the stiffness? And I think let's talk about that at the break, because number one, there's a lot of nuances with different donors and things. And I'm sorry I didn't talk about things that we've discovered, because I did want to. These questions are really hard, and we want to get tools out to people who will use them, because then they'll have bandwidth to do the large N that people need, because there is a lot of donor variation, and we need a large N to really learn things.
Brilliant, thank you. And Linda will be available for more questions.
I'm staying here till Friday.
Brilliant. Okay, thank
Not
you very
here,
much.
but in Cambridge.
Brilliant.